I'm going to try and convince you on a couple of things tonight, but I just need to apologise in advance that despite everyone giving really good talks tonight, if just click on, my job title is the best chat line on the planet, and to justify this I'm just going to go through the standard protocol in a pub. So it'll be the conversation of, I'm a microbiologist. Woo! Exactly. And then it goes on to, I'm a fungal microbiologist. Woo! Yeah, well, this scientific audience is getting in with it, but a normal, everyday person in general, like Joe Public or Jessica Public, whatever, you can see that the heat is starting to descend at this point, and you have to reach for some sort of point that they might understand what you're talking about. And I tend to accidentally go on to, have you ever heard of athlete's foot or thrush? Conversation over. And I've been doing my entire crew a miscredit. And if you skip on, please. So yeah, as I've said, millions of people suffer superficial infections such as athlete's foot, thrush, ringworm, you name it, funky toes, go for it. But fungal diseases kill more people every year than malaria or tuberculosis, so it's hella important. And also, if you do actually suffer an infection in your bloodstream, there's a high mortality rate of around 50% because of a high level of resistance and not many people looking for drug targets within these fungal cells. It's gone quiet, you're taking it seriously now. Yeah, get the section. Skip on, please. Skip. So the fungus that I look at is Candida albicans. Every year there's around 400,000 life-threatening infections from it. And it's got sort of two main growth types. It's got the yeast form up there. So if you've heard of yeast infections and stuff like that, yeast form. And it's also got this more root-like structure in the bottom left of hyphae, and this is normally more associated with the disease due to this almost burrowing and penetrance of the hyphae itself. And did you skip 2 slides? You did?
Yeah.
Hey. Sorry, but yeah, so I work on Candida albicans, but I look at a particular point of the disease, or a specific part of the disease called nutritional immunity. So we as human beings require certain nutrients to live. So if you've got anemia, you take an iron tablet. If you're hungover, you take maybe a zinc tablet, get your alcohol dehydrogenase going. But organisms that infect us, they need these nutrients as well, and they can't go down to the shop. So they use us. They steal nutrients from us. And we've evolved together symbiotically and in disease. And we've got this complex relationship of give and take, where the fungus might be trying to steal nutrients from us, and we're saying, no, it's all ours. And so when the fungus goes into your bloodstream, it's a very barren place. There's not much nutrients available for it. So these are some of the mechanisms that I investigate. So if you skip on. There we go. So zinc's very important because around 10% of the proteins in your body require zinc to actually function, and I investigate the function of zinc. So, skip on. Cool. So the particular thing that I'm interested in the fungus itself are zinc transporters. So these are proteins in the cell boundaries, so on the cell surface or inside organelles, small compartments within the fungus, and these traffic and move the zinc about. So they're important for taking zinc from the environment into the cell. And they're also important for putting it into its pocket or whatever. If you skip on, please. So how do I investigate this very complex mechanism? The advantage of working with Candida albicans is I can use a process called genetic manipulation. So these zinc transporters are proteins are produced from genes, and we know the DNA sequencing, we can predict it, so we can go in, delete a bit of DNA, therefore no protein, and we can see what happens. It's a big playground of DNA and really, really messed up fungi. And I use this technique and I complement it with fluorescence microscopy. So we have fluorescent dyes which stain zinc and fluoresce. So when we look down a microscope, we can see where the zinc is in the cell. And we use this to complement the genetic mutations. So if we knock out a zinc transporter, which brings zinc into the cell, we could say, there's no more zinc. And we'll go on to see that. So if you skip the next slide, please. So if we consider a fungus, it's got its gene there, it's producing protein, it's great. Click, please. It's nice and happy. Hey! It's getting zinc inside it. And if you click again, if the red structures there are hyphae and fungal, so they're the fungal cell, and the blue is zinc. So cell has transporter, can take zinc into the cell, we can look down the microscope and see it. But what happens when we get rid of the gene? We get no more protein, we don't get any more zinc into the cell. Sad, folks. So this is just a very brief overview of what I do, and these are the two main mechanisms that I use to investigate an incredibly important set of diseases. So if you skip on. So yeah, I hope you've all learned a wee bit of something today. Fungal disease is incredibly important, but it's very underappreciated, so tell your friends. I use gene disruption and fluorescence microscopy as my main methods, and therefore, hopefully you've been slightly convinced that my job title is the best chat line in the world, and I'm not saying best for what. So if you skip on, thank you very much. Anyone got any questions?
So you said the bloodstream was like a desert.
Yes.
So what does yeast, what does Candida do to change it not from the desert? I mean, presumably it's doing something to try and get more zinc in.
So there's a few mechanisms that Candida has when it gets in. It can aggregate with the hyphal form, but it's got a very cool system called siderophore. So it secretes a protein which goes out into the bloodstream, binds zinc, and then that protein will reassociate with it and it can take it up. But it can also, there's some evidence that it might be able to affect our cells as well. But the intracellular stuff, so the zinc transporters inside the cell are very important, because if it's going from, say, our GI tract, where there's a lot of zinc, it's got a little backpack full of nutrients in its vacuole, and when it goes into the bloodstream, it goes, there's no more zinc in here, crap, I need to use this stuff in my backpack, and starts transporting and being able to use it then. So these are some of the things I'm interested in. Cool. Fair enough. Thank you very much.